Hello and welcome guys. Now today we are discussing about the process of protein synthesis called translation. Translation means it is the process of conversion of RNA into proteins. This process actually takes place in 4 steps. The first step is called charging of tRNA. The second step is initiation. The third step elongation, and the fourth one is termination. During charging of tRNA with amino acids, it is a two-step process taking place or catalyzed by an enzyme called amino acyl tRNA synthetase. In the very first step, amino acid is charged or activated by addition of AMP from ATP. Subsequent hydrolysis of pyrophosphate makes this reaction irreversible. In the second step, amino acid is transferred to the 3' OH group of tRNA by an ester bond when AMP is displaced by tRNA, creating an amino acyl tRNA. Amino acyl tRNA is then released from the enzyme. This is known as formyl methionine tRNA, which will initiate the protein synthesis. Now this will recognize the initiation codon AUG. AUG means it recognizes the amino acid methionine. Now coming to the second step called initiation. Now as we know that translation occurs in the cytoplasm where ribosomes are present. Now initiation involves binding of smaller subunit of ribosome carrying amino acyl tRNA to the 5' end of mRNA with the help of initiation factor IF and some other proteins which help in this reaction, as you can see over here. It simultaneously binds to peptidyl site of the large subunit of ribosome with the help of divalent magnesium cation Mg2+. Now next step is elongation. Elongation occurs when next amino acyl tRNA which is charged, which is in line at the A site or amino acyl site along with GTP and an additional elongation factor EF. The chain goes on elongating further, producing polypeptide with the help of peptidyl transferase. Finally, we arrive at termination. The termination of polypeptide synthesis occurs here in the picture. It occurs when A site of ribosome notices nonsense codons like UAA, UAG or UGA, which no tRNA is able to recognize. In this process,